Assalamu alaikum, dear students. I am Muhammad Rauf. Welcome to the course Principle in Animal Life 2. This is lecture number 20. The topic is chromosomal mutations. Discussing agenda for the topic is alteration of chromosome number or structure causes some genetic disorders, human disorders due to chromosomal alteration, change in number of chromosome, change in number of chromosome, aberrant euploidy, alteration of chromosome structure, aneuploidy of sex chromosome, Down syndrome. Lecture outcome. After watching and listening this lecture, students will know about various types of mutations in chromosomes and the symptoms associated with them. Alteration of chromosome number or structure causes some genetic disorders. The phenotype of an organism can be affected by small-scale changes involving individual genes. Random mutations are the source of all new alleles, which can lead to new phenotypic traits. Large-scale chromosome changes can also affect an organism's phenotype. Now, mutations are changes in the genetic material of a cell or an organism. Mutations may be gene mutations where change occur in the nucleotide sequence of a particular gene, or chromosomal mutations, which are large-scale mutations in which a chromosome having several genes are involved. Either duplication of chromosome occurs or deletion or translocation. So they are usually large-scale mutations, and quite often they are so detrimental that the individual cannot even make it till birth and usually dies during development, during embryonic development, or results in miscarriage or stillbirth. So they have severe consequences on the phenotype because the chromosomes consist of DNA and associated proteins, and DNA carry genes. The genes carry instructions for the development of the organism, for controlling its different traits, for the synthesis of proteins, enzymes. So such large-scale mutations usually are detrimental and they have drastic effect on the organism's phenotype. Physical and chemical disturbances as well as errors during meiosis can damage chromosomes in major ways or alter their number in a cell. We have already discussed the process of meiosis and mitosis, where the chromosomes are arranged on the metaphase plate, and then the spindle fibers arises from each end, and they attach to the kinetochore located on the centromeric region of each chromosome, and then they start to depolymerize. The spindle fiber starts depolymerization, they are shortened, and in this way they drag the chromosomes towards their respective ends. So sometime problem occurs during this during this process. The metaphase plate is formed and the spindle fibers are not properly attached to the uh, chromosomes on their respective ends. As a result, sometimes both the members of a homologous pair enters one gamete and the other gamete completely lack any member of that particular chromosome pair. So such type of instances do occur during meiosis, during gametogenesis. Large-scale chromosomal alteration in humans and other mammals often lead to spontaneous abortion. Spontaneous abortion means abortion at once. An individual born with these type of genetic defects commonly exhibit various developmental disorders because the process of development is so much complicated. There are so many genes and almost all chromosomes have genes which play some role in this developmental process. So any kind of chromosomal mutation will have effects on development. Plants appear to tolerate such genetic defects better than animals do. Yes, it's a factor because the triploidy, tetraploidy, hexaploidy, it is much common in plants as compared to animals. So they are in a better position to tolerate such changes. Now here we will give an overview of the chromosomal mutations. So there are two broad types of chromosomal mutations: change in the number of chromosome and change in the structure of chromosome. Change in number of chromosome can be of two further types, and it's aberrant euploidy and aberrant aneuploidy or aneuploidy itself. Aberrant euploidy means that each organism has a particular chromosome set 2n, and the 2n number can be 46 in humans and 8 in Drosophila and 78 in dog. So when a complete chromosomal set is added, then this is called aberrant euploidy. Suppose a human sperm has n number of chromosomes and a human ovum has n number of chromosomes. When they fuse together, they form 2n, 46 chromosomes. But what will happen if two sperms fertilizes an egg at a time? Now the chromosome complement of such a situation will be 3n. So one complete chromosome set has been added and the normal number has been disrupted. Now such an egg fertilized by two sperms will have 3n chromosome number. 3n, and such an egg will not survive. Aberrant aneuploidy means when one or two or three chromosomes are either uh, missing or they are more than the normal. So the difference between these two is that here a complete chromosome set is either missing or extra, and here only the individual chromosome either is missing or extra. So aneuploidy may be monosomy. Monosomy is a condition in which one of the chromosome is missing. Just as we said earlier that during embryonic development, during meiosis. Sometimes both the members of a homologous pair enters one gamete. So in such cases, two gametes will be formed. One will have 22 chromosomes, the other will have 24 chromosomes. So a gamete having 22 chromosomes is fertilized by a normal sperm. So the chromosome complement of that fertilized egg will be 45, 2n minus 1. So this is called monosomy. The second is trisomy. Trisomy is a situation where the other gamete, which had 24 chromosomes, is fertilized by a normal sperm. So the overall chromosome complement of the a fertilized egg will be 47. So this is called trisomy. So you can describe monosomy as 2n minus 1 condition and trisomy as 2n plus 1 condition. And of course disomy is normal. Now monosomy, now one chromosome is missing here. It is 2n minus 1 situation. But that one missing chromosome can be a sex chromosome or it can be an autosome. Similarly, the one chromosome which is an extra copy can be an autosome or it can be a sex chromosome. And here change in the structure of chromosome, it can be deletion, duplication, inversion, and translocation. Now change in number of chromosomes, aneuploidy. When the chromosome number is above the normal is 46, below normal 45, or above normal 47. So it is called aneuploidy. There is an occasional mishap called a non-disjunction during meiosis. Non-disjunction is the failure of the homologous chromosomes to separate from each other. The chromosomes join end-to-end, homologous chromosomes join end-to-end with each other during prophase, and there they undergo a process called crossing over, where exchange of segments takes place, and then these duplicated chromosomes are arranged on the metaphase plate, and sometimes the duplicated chromosomes, the individual members are unable to separate from each other, and both of them enters one gamete, and the other gamete completely lacks a copy of that particular chromosomes. So in non-disjunction, one gamete receives two of the same type of chromosomes and another gamete receives no copy. If either of the aberrant gametes unite with a normal one at fertilization, each of the aberrant gametes, means the one which has 22 chromosomes and the other which has 24 chromosomes. So if they combine with a normal sperm, then in one instance there will be 2n plus 1 situation, 47 chromosomes, and in other instance there will be 2n minus 1 situation and 45 chromosomes. The aneuploid zygote is monosomic if it is lacking one chromosome from its normal number. Means if its chromosome complement is 45, just like we have in Turner syndrome. If a chromosome is present in triplicate in the zygote, so that the cell has 2n+1 chromosome, the aneuploid cell is called trisomic. Trisomic, usually the Down syndrome, where one somatic chromosome is in excess, and Klinefelter syndrome, where one sex chromosome is in excess, is a 2n+1 situation. Mitosis will subsequently transmit the anomaly to all embryonic cells. So if the chromosome complement of a zygote is 2n-1 or 2n+1, then all the descendants of that zygote will have the same chromosome complement. Change in number of chromosome continues. So monosomy and trisomy are estimated to occur in 10 to 25% of human conceptions. Means there is a probability of 10 to 25% in every conception that such an error will occur, and it usually results in pregnancy loss. Means the pregnancy is lost because even gene mutations are so harmful that they result in cancer and eventual death of the organism. So just imagine the chromosomal mutations where several genes are involved. So chromosomal mutations are harmful, they are lethal, and they result in pregnancy loss. Even if the organism survives, then there are several abnormalities linked with that person. Some organisms have more than two complete chromosome sets in all somatic cells. Now we are talking about aberrant euploidy. Euploidy is the chromosome complement, the number of sets in a cell or an organism. So if the change occurs in the number of sets, then it is called aberrant euploidy. The general term for this chromosomal alteration is polyploidy, means the suppose an individual is diploid normally, and then uh, the offsprings of that individual undergo some kind of mutation during the process of fertilization, and a triploid individual is produced, two sperms fertilizing a single egg, or a diploid sperm is produced. So in that situation, these are referred as polyploidy, means the multiples of the basic chromosome complement, triploid 3n, tetraploid 4n. So one way a triploid cell may arise is by fertilization of an abnormal diploid egg produced by non-disjunction of all its chromosomes. Diploid egg may be produced where all the chromosomes located on metaphase plate ends in one cell and the other cell completely lacks any of the chromosomes. So if such a diploid egg is fertilized by a sperm, the result will be a triploid cell. Tetraploidy could result from the failure of a 2n zygote to divide after replicating its chromosomes. Subsequent normal mitotic divisions would then produce a foreign embryo. Polyploidy is fairly common in plant kingdom. It's more common. Plants are more tolerant to that situation, whereas animals, if such situation occurs in an animal embryo, it is usually discarded through miscarriage or spontaneous abortion. The spontaneous origin of polyploid individuals play an important role in plant evolution. Polyploidy evolution is a very slow process. It requires hundreds of thousands of years, but polyploidy is a phenomena in plants where a new species can evolve within 2 generations, within 1 generation or within 2 generations. Amazing. Many species we eat are polyploid, like bananas are triploid, wheat hexaploidy, and strawberries octaploidy. Polyploid animal species are much less common, but there are few fish and amphibians known to be polyploid. In general, polyploids are more nearly normal in appearance than aneuploid. Means polyploids have a normal appearance even though they have extra set of chromosomes, but their appearance with normal individuals is much more as compared to aneuploids. One extra or missing chromosome apparently disrupts genetic balance more than does an entire set of chromosomes. Now, it is an interesting statement. They are saying that if one chromosome is missing or extra, its effect on the genetic balance is much more as compared to if a complete chromosome set is missing or extra. Now here we will analyze the process of meiosis, how the 2n minus 1 and 2n plus 1 eggs are produced. So these are two different cells. In one of the cells, they have shown the process of non-disjunction, which results in two types of gametes. Two gametes that are having an extra chromosome and two gametes that are lacking a chromosome. So this is the starting cell and it has 3 chromosomes. Then those 3 chromosomes become duplicated and then they are separated in the anaphase 1, and this cell completely lacks that.